we've arrived at part 6 of the cell and the associated ideas about the cell, including plasma membrane and organelles. And here we have what could be described as a typical looking cell, although cells take many shapes and formats in order to accomplish their mission. So there are hundreds of tissue types employing cells of a variety of morphological qualities. Some can be very long, like the cell that it represents the individual cell in a muscle that runs the length of a thigh muscle, right? And so that's a gross anatomy length, but it's being accomplished by a microscopic morphology. So you can't actually see the individual muscle, despite the fact that it's traveling and its contractile portion is traveling several inches in length. Kind of an interesting detail. Even more so would be nerves or neurons within nerves, in which the neuron itself is microscopic, and yet it can travel from the tip of your toe all the way to your lower back as an individual cell, right? And there's a cell body, so to speak, with a nucleus. So what I want to start here with is just the cell in general in its situ. So one of the things that we do in cell biology is that we conduct experiments regarding receptors. We can find ways to bind to receptors. We can use fluors, which light up the, how shall I say, monoclonal antibody that is binding to the receptor on the cell. So you can imagine looking through your microscope, and what you see is oftentimes a glowing green colored fluor. And as you observe through the microscope, it's literally moving across. It has bound to a receptor. In some cases, it's done something that we call patching, which is to say it's pulled together some of these receptors into large groups of fluors, and it's moving toward a pole. So cells tend to have poles. There's the pole in which the membrane is being reabsorbed, and so then the fluors get pulled within the cell. Okay, and then the cell will, how shall I say, replenish the cell membrane, but meanwhile the cell membrane is continuous with much of the cell membrane within the cell, and then it goes, you look over at the other pore, the other pole, I should say, of the cell, and you have new membrane coming out. Now, one of the deals that you can imagine is that the cell can adjust its receptors. It can adjust its concentration and number of receptors per unit area. Okay, so that if you were to introduce a drug, say some kind of a drug that relieves pain, so an opiate, that you can actually affect the analgesics of neural tissue and cause inhibition. There's a variety of ways to do that we'll take a look at in the neuroscience portion of our course in the peripheral nervous system. But at any rate, before we get to that point, let me just say that it's possible to introduce exogenously, and I want to use the term exogenous versus endogenous. Okay, so endogenous is what the body does. So endogenous, let's write that down, endogenous versus exogenous. Exogenous is what we introduce into the body from the outside. We need some exogeny. We need nutrients. We need substrates. And there are some essential nutrients. And I've given you a list of those things just to remind you. I had a listing of 7 items, and they begin with oxygen, water, I don't see it here at the moment, but oxygen, water, glucose, and amino acids, omega-3 and omega-6 fatty acids, vitamins, minerals. These are absolute requirements. You cannot live without these substrates for the manufacture of both structure as well as working molecules within the cell. So that's the essential exogeny, and then we add other things. You know, anything from pizza to antibiotics would be, how shall I say, extra exogeny. Endogeny is something that we consider from the point of view of physiology. Physiological, how shall I say, synchronicities and balances and uninhibited flow are required for healthy homeostasis. So we go to bed on time, get up on time, we breathe correctly. Some people actually study breath, both in terms of meditation as well as in the choreography of dance, as well as in opera. Learning how to use breath and voice, for instance, is considered an art form. So I would say endogeny is very important to us. There are daily practices that make our life better, whether they are exercise, sleep, a proper diet, and so on. Those are all things that we balance from within. So I want that clear. There's endogeny, there's exogeny. One of the difficulties of doing research is that in order to measure something, we have to push a tissue hard enough or an organism hard enough in order to result in something measurable. And as a result, a lot of our pharmacological measurements are made exogenously in ways that do not necessarily represent physiology, the endogeny of physiology. Right? So there are differences that occur. It's different if you take the blood pressure of somebody lying down compared to standing up, walking, and so on. So the heart has to work a little bit differently under different conditions. And that's a normal endogeny that can be more or less measured from the outside. Okay, I want to bring all of this to your attention because there's such a thing as addiction, which is a huge thing in our own, how shall I say, social and scientific and medical universe. And so imagine that you have introduced an opiate to a cell. Now it turns out that opiates are something that the body itself uses. It can be very powerful, like endorphins that are part of the central nervous system, and it can be somewhat mild analgesics like enkephalins, which might reside closer to joints and muscle attachments and so on. So from the mild to the wild, we do have analgesics within our body, and so we utilize that when we add something from the outside, an exogenously introduced molecule. So the question is, if I were to actually sequence an endorphin, an opiate, which is the perfect endorphin, stronger, more, how shall I say, powerful than heroin or morphine. It's precisely the molecule that the body itself uses to bind to its own opiate receptors. And I were to introduce that body-specific endorphin, would the cell have something that we call receptor down syndrome? The idea being that when we introduce opiates, the cells tend to withdraw receptors. The receptors become fewer and fewer per square inch, so to speak, or per square area on the cell surface. And so as a result, we need to add more opiates sometimes in order to get the same result. And so basically what the cell is doing is it's saying, this is not something that I've made the arrangement for, therefore it's not supposed to be here, and I'm withdrawing my receptors. And the thing is if you were to even make a perfect receptor or a perfect molecule, an endorphin that is identical to the human body's own opiates, then in fact it would even be more profound, the withdrawal, because the cell can actually recognize that this is stuff that is made within the body, and I did not make the arrangement for this. And so I'm sort of anthropomorphizing individual cells here, but nevertheless I'm pointing out that cells will reduce their receptors. There will be receptor down syndrome as the cell actually, how shall I say, reintroduces its own membrane and then brings it back out again on the surface. And it's constantly circulating membrane. And in that way, it changes the amount of receptors on the surface for various things.